everyone, I'm going to go over the results on an experiment conducted to study the effects of lychee seed extract on HL60 cells. Great efforts have been made for decades to try to find cures for different types of cancer. One route of research has been to study a line of human leukemia cells known as HL60 cells. Normally they differentiate into granulocytes or monocytes. But when they are deemed cancerous, they remain undifferentiated in the myeloid progenitor phase and continue dividing. Scientists and researchers have been trying to find a way to eliminate the problem of these endlessly dividing HL60 cells. One plan of action has been to induce apoptosis in these human leukemia cells via oxidative stress. Apoptosis is the deliberate activation or inactivation of specific genes to initiate the process of programmed cell death. Apoptosis is visually characterized by cell shrinkage, nuclear condensation, and DNA fragmentation. In this process, the plasma membrane of the cell does not rupture. Rather, it stays intact so that none of the contents of the cell leak out. One possible way to force HL60 cells into apoptosis is to introduce them to polyphenols, a plant secondary metabolite. In certain amounts, plant polyphenols can cause a cell to go through programmed cell death. However, introducing the cell to too much could cause it to go through necrosis instead, since it puts a lot of stress on the cell. Necrosis is a version of cell death that usually occurs when the cell is exposed to high stress. In this case, the cell swells and lyses, causing the contents of the cell to spill, which also puts stress on the surrounding cells. Many plant polyphenols have been shown to have anti-cancer properties. In this experiment, the effects of lychee seed extract were tested on their ability to induce apoptosis in HL60 cells. However, this is not the first time lychee has been tested for its anti-cancer properties. In a previous study, its effects on breast cancer were explored, which yielded positive results. The experiment was conducted on mice with human breast infiltrating duct carcinoma. The mice were treated with lychee fruit extract, and the results showed that there was hindered growth in the breast cancer cells and a reduction in tumor mass. The researchers attributed these results to a couple of different attributes of the fruit, the primary one being the ability of the plant's polyphenols to help induce apoptosis and decrease cell proliferation. For this reason, lychee was chosen for this experiment in hopes of producing similar results. In this study, a viability assay of the HL60 cells treated with lychee seed extract was conducted using WST8. WST8 is a substrate that turns orange when it is reduced by cells, and only viable cells can reduce it. Therefore, an increase in absorbance at 450 nanometers shows the presence of viable cells. Figure 1 shows the average absorbances of the WST8 colorimetric viability assay on HL60 cells. The bar labeled control represents the average absorbance for wells containing only HL60 cells in media. The bar labeled 70% ET represents the average absorbance of wells containing cells, media, and 70% ethanol, which was used to extract the lychee seed. The third bar represents the average absorbances for wells containing cells, media, and camptothecin, which is a known apoptosis inducer. The last bar represents the absorbances for wells containing cells, media, and lychee treatment, which was extracted with 70% ethanol. Unfortunately, when the average absorbance for the control groups containing untreated HL60 cells was compared to the average absorbance of the lychee-treated HL60 cells, there was not a significant difference in viability. This can be seen in Table 1 in the t-test between the two, which yielded a p-value greater than 0.05, indicating no significant difference. The only significant difference found was between the untreated cells and the positive control group containing camptothecin. A fluorescence microscopy assay was also done in order to see if the HL60 cells were affected by the lychee treatment, and if so, in what way, either apoptosis or necrosis. The cells were stained with cyto16, a fluorescent dye that binds to DNA and makes it glow green. This allows nuclear condensation and blebbing to be seen under a confocal fluorescence microscope. The picture on the left shows untreated HL60 cells. As you can see, they are healthy in appearance in that there is no nuclear condensation or blubbing as there would be in apoptosis, or cell swelling and lysing as there would be in necrosis. The picture on the right shows cells treated with camptothecin. So these cells are going through apoptosis. And you can see nuclear condensation and blubbing. This figure shows HL60 cells that were treated with lychee seed extract. As you can see, the cells appear to be swollen, but there are no clear indications of either type of cell death occurring, which was also the conclusion drawn from the viability assay. The HL60 cells treated with lychee seed extract were also stained a second time. This time, the only dye used was oxidative stress detection reagent, which also fluoresces green. As the name implies, the cells were dyed with the OSDR to detect the presence of reactive oxygen species, or ROS. ROS are important to cells, but if they reach a concentration that is too high, it can cause the cell to go through apoptosis or even necrosis, depending on the level of stress. This figure shows the reactive oxygen species detected in the HL60 cells after 24-hour treatment of lychee seed extract. The final part of the experiment was a COMET assay, also called a single-cell gel electrophoresis, to test whether or not the lychee seed extract contained a polyphenol that could damage the DNA of the HL60 cells. The assay used Cy-green dye to show whether or not individual cells were damaged. Undamaged compact DNA moves slower in an electric current than broken or less condensed DNA fragments. Therefore, the head of the comet will not move nearly as fast towards the anode during electrophoresis. If there are any damaged or loose DNA fragments, they will travel faster, creating a tail-like structure that streaks away from the comet head towards the anode. The longer the tail, the more damaged the DNA is. The figure on the left is a reference piece that gives different classifications for different lengths of comet tails. On the right is the image taken from the microscope that shows some comets from the HL60 cells treated with lychee seed extract. There were 3 definite comets that could be identified, 2 of which were classified as NC0 and 1 which was classified as NC2. So generally speaking, no real DNA damage was detected. Based on the results to the 3-part experiment, it can be reasonably concluded that the lychee seed extract did not significantly affect the livelihood of the HL60 cells. It did not produce absorbances that suggested a significant difference in cell viability, nor did it appear to cause any definitive type of cell death in the microscopy assay. After examining the COMET assay, no significant DNA damage was found. A future direction for this project could be to increase the amount of lychee seed extract that the HL60 cells are treated with. The experiment on the effects of lychee treatment for breast cancer previously mentioned was dose-dependent. Small amounts of lychee fruit treatment did not affect cell growth or tumor mass in mice like larger amounts did. Perhaps that is the case here. Maybe an increase in lychee seed extract would produce different results.